who's a clinician scientist. Oh, sorry, we're recording. I'll start again. Very pleased to introduce Dr. Martin Kaiser, who's a clinician scientist at the Institute for Cancer Research, where he leads the myeloma molecular therapy group. He's a consultant haematologist at the Royal Marsden, and we work very closely with Martin on a number of myeloma trials here at Leeds CTRU. So today I'm really delighted to welcome him to talk to us about the MUK9 Optimum trial, looking at first-line treatment for patients with high-risk multiple myeloma. And these results were presented at ASH, and he's going to talk us through them today. So welcome, Martin. Just usual T's and C's for a Zoom conference. We'll take questions at the end, so if you keep your mic on silent, that would be great. We can take questions via the raise hand function or via chat. But I'll hand over to Martin for a presentation on MUK9. Thank you.
Thank you very much. Thank you very much, Sarah, for the very kind introductory words. Leaves little for me to say, but to actually start, I think, straight into the talk. And let me see whether I can share my slides here. Probably if you can just give me a brief verbal heads up that
yeah, we can see those. Thanks, Martin.
Okay, great. Thank you very much. And yes, I have, of course, the pleasure to present the work that we first of all presented at ASH and are currently looking at publishing as well to a wider audience on the primary endpoint result of the Optimum trial, which is really a huge collaborative effort with Sarah and her team and really quite a number of researchers across the country, but also some, really feeding into collaboration internationally, and especially positive, as we will see, the trial actually has a positive readout. So the title is very bulky. Maybe I'll actually just dive into the talk instead of making this too complicated. So the MUK9 trial, these are, the ASH slides really, and this is normally what we present. There is disclosures as a start, but then coming to the aim of the MUK9 trial, the Optimum MUK9 trial was a trial that we designed. Now probably the first design discussions that we had are dating back 6 or 7 years into 2015, 2016, because it was very obvious then already, and I think it has only become more obvious since then, that there is really a high unmet need in a group of patients that isn't really too small. And this is exemplified here on the left-hand side slide. We do normally treat patients with newly diagnosed myeloma, especially younger patients with quite intensive first-line treatment, including autologous stem cell transplant, which many of you will have heard already. But we're keeping normally that treatment relatively the same for the patients, regardless of any features maybe that we are finding. Of course, we're trying to personalize medicine in the way if we know that there are pre-existing conditions or side effects that make the tolerability of treatment worse for some people. But other than that, we don't really have any means also on the NHS to adjust treatment. And there aren't really international guidelines either that would be suitable in telling us to treat patients specifically depending on certain features. But we do know that if we do that, 20 to 25% of patients unfortunately experience a relapse of their disease, often within the first 2 years of follow-up after transplant. And that means, of course, these patients are then presenting with, as we call it, clinical high-risk disease. So they have, the disease just has shown through the afterwards experience of early relapse that it is probably behaving more aggressively than other types of disease. And we can, of course, then try to adjust treatments. Luckily, we have had an increase in availability of relapse regimens for myeloma, but it still remains that, unfortunately, this group historically has shown a reduced response to second-line therapy. These patients experience stress by being told a range of outcomes at the beginning when a disease is diagnosed and then experiencing that the disease is coming back earlier than maybe for many other patients. It can be a strain on resources. Actually, maybe it's not the right expression. What we're trying to say is we are keeping every patient under close checks because we are struggling so much to predict at the moment who is relapsing early, whereas some patients may actually be able to stay away from clinics after a transplant for longer because the disease is not falling into this bracket. And there is a insight that we're gaining from laboratory research that is, I think, now probably become more commonplace because of what everyone has experienced more now through media with COVID. We know with COVID, if there's more COVID around in the world, there's probably more likelihood that one of the COVID strains gathers or gains a new mutation, which may make its properties different. And I think the same is something that has been at least postulated in tumor evolution. If there is more tumor around, each of these cells can potentially undergo a mutation, and there could be a fast tumor evolution if we wait for the disease first to relapse before we act, if we find it to be aggressive. And the aim is here shown on the right-hand side is really that we actually use the insights that we have gained through our research over the years in myeloma to use biological markers to improve biological risk prediction, which could help us to actually apply extended diagnostics at the diagnosis of the disease, identify those that are likely to relapse or have a high chance of relapsing early, and actually to modify the approach of treatment early, which means that we adjust the resources as well to the unmet need of these patients. And we can hopefully empower patients by knowing more about their disease from the start. Now, this is a very technical slide, and I think I'm mindful that it's a very heterogeneous background of people that are people with very different backgrounds that are joining us on the call today. So this slide just explains that there are different tools, diagnostic tools that we can apply when a patient gets diagnosed that can tell us more about the risk of an early relapse, probably than what we are using at the moment in the clinic as an approved standard of care diagnostics. On the left-hand side, we see genetics, and to be fair, the UK myeloma research community and UK patients have contributed massively to establishing this model on the left-hand side, which we are already using in standard of care now in most sites, and we're trying to improve it through a currently ongoing trial, the RADAR trial, to actually get profiling for all of these genetic lesions that you can see here on the left-hand side established. And by profiling all of these lesions, which has not been standard of care has been only profiling these upper two type of abnormalities here. But by profiling all of them, it allows actually to count how many of these lesions, these are tumor genetic lesions, so aberrations that the tumor cells, the myeloma cells have acquired in the development of the disease. And they have all individually been known to be associated with adverse outcome. But actually, if the two or more of these co-occur in one tumor cell, we do know that unfortunately the disease is at really high risk of early relapse. And this has to do with that the tumor is genomically unstable and probably is more prone to acquiring new mutations relatively quickly. Then there is a genetic analysis tool or a genomic analysis tool called gene expression profiling, which has been around and particularly was a focus of research about 10, 15 years ago. But as a result of that, diagnostic tools have been developed that also can help identify patients that have a high risk of early relapse with a so-called gene expression high-risk signature. Sky92, for example, is one that is now fitting the very stringent and strict criteria for diagnostic tools that can be applied in standard care. It is not applied widely because it is currently not reimbursed in the NHS, and actually, to be fair, at the moment, also not widely in any other healthcare system, but this is something we are working very actively on now, which we can discuss later to actually make this more widely accessible, because it identifies other patients than those that are just identified by genetics. There's a certain overlap, but there is about 10% of patients that can only be identified by gene expression profiling. And altogether, having one of the two type of risk markers identifies about 25% of patients. That about number of patients that we also find when we just look retrospectively how many have relapsed early. So really fitting the experience in clinical practice. And then there is an entity that is relatively rarer than the other ones, which is called primary plasma cell leukemia, which is a disease type whereby the myeloma cells are growing in the peripheral blood, which can already show that they're more aggressive, and they're also quite proliferative. And they have very similar properties as these other two high-risk markers. So really the commonality across these three entities is that there is, with current conventional therapy, relapse of the disease on average about 2 years, within 2 years from start of treatment. This is what, in the purposes of this research, we have termed ultra-high risk disease. Now, this is, of course, a term that can mean different things for different people. I think we had internationally to make clear that we're really trying to address the highest unmet need here of patients, because there is a very wide definition of high-risk disease, and sometimes to the extent that the definition of it gets blurred. So we were actually trying to define with this term the fact that this is the group that has a very high likelihood of relapse within 2 years from start of treatment, to make it clear that this is what we are meaning. And all these can actually be accessed diagnostically, even if they're not standard of care accessible tools necessarily. Now, I think some of you might be wondering, why hasn't research already focused traditionally on improving the outcomes of patients with such high-risk disease? And we have had some insights from trials that were actually including all types of patients, and retrospectively it was then looked at whether treatments were maybe more beneficial for patients with high-risk features or not. But to make really progress for this group, which is only a quarter of all patients, we normally had to wait for the trial results for every patient, or all the patients, the majority of the patients, to actually mature. That means these patients ultimately had to wait for longer, proportionately, until we could make a statement only retrospectively about whether a certain therapy has benefited them more than another one. And this is really something that we felt clearly needs to change. So we wanted to design a trial that really investigated whether a specific treatment was benefiting patients with high-risk disease. But the problem came up in the design considerations some 5 or 6 years ago when we started, that if you randomize these patients, this is a typical method for clinical trial research, of course, whereby you by chance have to allocate a patient to the one or the other treatment, one of which has to be the current standard of care. It is difficult to do that really ethically for patients, because we have to inform the patient then that they have this ultra-high risk or high-risk disease, but at the same time there could be a one-to-one chance that you might get a treatment that we know already doesn't work well for you. So this is a challenge. You know, everyone feels that this is not the right way to do research, and I think purely practically as well, it is likely difficult to successfully recruit to such a trial. So this is really where Sarah's team came in with, brilliant methodology that has been developed especially to address the needs, unmet needs of rare disease groups. And I think that's what we're really talking about. Myeloma as such is not a very frequent disease. Now we're looking at only a quarter of the patients that have these specific markers. This is really a rare disease group. And for these purposes, from other diseases that were even rarer, in this a method that actually compares the outcome of the new treatment against a very defined group of patients that have received standard of care treatment, but in another trial, which we termed a digital comparator trial, was something that Sarah's team came up with and that we were really able to adopt here. Now, the important thing is that it has to be predefined what it is compared against. The comparison, of course, then means we can tell every patient that signs up to the new research that they all will get the new experimental therapy, which we at least have designed in the hope that it will benefit patients. And this makes it, of course, a patient-centric approach. It also makes use of the existing resources very effectively and can lead to faster results, because we only need to enroll half the number of patients into the research that is investigating the treatment. But of course, it depends very much what you compare against. So it has to be carefully designed so that your standard of care group does not look ultimately an unfair comparison against your treatment. And this was something where really we stuck our heads together, both coming from Sarah's team side in terms of statistical work and our team side from the genetic analysis side to come up with a design that is as precise as possible in its comparison. It also is a technicality that such an approach uses a different type of statistics, a Bayesian framework, which is different to standard types of statistics that is used in most comparative trials. Now, this is the design that we actually used in this trial. So in the top part, you see the Optimum trial, which is the treatment that the patients received that participated in the new research. And I will come to the exact combination in a second. And the comparator is a trial that was also run in the UK and was only possible through the contribution of patients in the UK, the MYELOMA-11 trial, which was the largest trial run so far for myeloma, with 4,400 patients participating overall. And we were actually going to select only the treatments that were showing the best outcomes from the MYELOMA-11 trial, the KCRD or CRD induction treatment, and actually only those patients that also had high-risk disease. And the difficulty here was that the outcomes of the Optimum trial, the MYELOMA-11 trial, weren't even analyzed yet at the time when we were designing this trial. So we had to, on the one hand side, of course, pre-plan for that, but on the other hand, I think it added to the stringency of our result in the end, that we took results on board that weren't even known yet. So this is the design and the numbers of patients that were participating in the trials. The Optimum trial, 472 patients with newly diagnosed myeloma fit for intensive therapy from 39 hospitals were screened and offered genetic and gene expression profiling. And those that were found to have ultra-high risk disease or plasma cell leukemia, as per our definition mentioned before, were offered participation then in the interventional trial with the drug regimen I will show in a second. These were 107 patients. And in the MYELOMA-11 trial, where we had stored material, we actually performed effectively simultaneous profiling of a larger group of patients that had been treated already on the trial, but we actually identified them only on the basis of the genetic and gene expression parameters I just mentioned, to actually construct effectively our comparator group of high-risk patients that were treated on the winning arms of the trial. So these were independently identified groups, and both trials recruited in the same area, of course, and used very similar trial entry criteria as such. So we were pretty certain that we were not excluding patients just by the barriers that we were defining by entry criteria in terms of high-risk disease. So this is a treatment that patients received on Optimum, a novel combination that hadn't at the time been really tested in trials before. And this was very fortunate, really, that we could get access, especially to Daratumumab in first-line treatment. It was one of the first trials internationally, globally, that were able to offer patients such a therapy. So this is Daratumumab, Cyclophosphamide, Velcade, Revlimid, and Dexamethasone as induction. Then a single autologous stem cell transplant together with Velcade, a consolidation treatment with Daratumumab VRD for 6 cycles, then a further consolidation with Dara VR, and maintenance until progression. And that is in contrast, maybe to just go 2 cycles back here, in at the time, definitely when the trial was designed, this treatment wasn't even available on the NHS yet, but had been just tested at MYELOMA-11. KCRD, so carfilzomib, cyclophosphamide, Revlimid, dexamethasone, a single transplant, and Revlimid maintenance therapy as the comparator in MYELOMA-11. Several other tests were done. We were looking in the evaluation at the efficacy of this treatment in comparison at 18 months progression-free time against the MYELOMA-11 treated patients. We also looked at response and minimal residual disease after induction therapy and the transplant. And of course, we were aiming to determine how safe this treatment is and which side effects are most common. So I'll probably just summarize this slide as this is a typical slide for most trials of 107 patients. Several patients did, unfortunately, either due to poor tolerance of the therapy or other reasons, not complete the complete treatment until the 18 months progression-free survival time point. But it were 80 patients, which is quite a high fraction, especially considering high-risk groups that completed consolidation 1 at the very bottom of this slide successfully. And this left for us with 4 patients having missing follow-up data, 103 to be included in the primary analysis population. And this primary analysis population, we then compared effectively against the population that we had identified only by the genetic screening parameters from the MUK9 trial. And this was a big uncertainty at the time of the analysis. Will we really have identified a group that is comparable and has similar characteristics in the new trial compared to the old one? And actually, we did. If you look at the different parameters that are listed here, including clinical parameters, age, ISS stage, and eco performance stages, these are very, very similar. And also the molecular profiles are very similar between the two groups, which actually really shows that the biological criteria that we define, the genetics that we define, are very stable characteristics. MUK9 recruited several years before Optimum, so we do know this is an overall global characteristic of the disease, that we do find a certain proportion of patients having either these double hit genetics or co-occurrence of 2 or more high-risk markers, or a certain proportion having the high-risk signature. And this makes the two groups really comparable here. And this is the primary trial endpoint, which I'm sure Sarah can probably comment on much better than I do, but it shows effectively how many patients are progression-free and alive after these 18 months from start of treatment. These are density distributions. So effectively, if you look at the peak of the density curve here, on the blue curve is the curve for the Optimum trial. So somewhat over 80% effectively of patients or around 80% of patients are progression-free and alive at 18 months. And that's a far lower proportion here, only close between 60 and 70% of patients on the MUK9 trial. And as per the statistical model, the Bayesian statistical model, that resulted in a 99.5% chance that Optimum trial treatment is better than the MUK9 trial treatment, which means the overlap of the curves was actually really small. There is very little doubt that these curves are showing a different result. I think that's optically already very clear. These are actually typical survival Kaplan-Meier curves, which show the same result, although in a different way of plotting them. And you can see here the 18 months survival median estimate is 81.7% for the Optimum treated patients, and 65.9% for the patients on MUK11. Now, these results are consistent. Patients on MUK11 receive 2 different types of intensity induction therapy, of which the KCRD treatment at the very end, which we didn't know when we designed the trial yet, turned out to yet lead to better results for the overall patient population. But as we can see here, for this group of patients, where the definition is double hit and or a gene expression high risk, the outcome is still significantly better if they're getting the new treatment in the Optimum trial, which is a very reassuring finding, of course. We looked at response as well. So we did see that the majority of patients reached a complete remission at the end of consolidation 1, 68%, and a striking 22% very good partial response, with only very few patients having a lesser response. And that correlates very nicely with what we have seen as an early response assessment. So the end of consolidation 1 was yet further 6 months after the transplant, actually 9 months after the transplant. We looked 3 months after the transplant at minimal residual disease in the bone marrow. We found a very similar minimal residual disease negative rate. And these patients all turned out in the end, more or less, to also end up having a complete response. So we could actually see a very nice correlation with patients turning MRD negative after transplant to then also being in a complete response. Later down the line, it took longer for some patients. We know that the serum parameters take some time to clear from the blood, which makes it harder to achieve a complete response early. but we see a very good consolidation of that through the treatment that patients received on the trial. Now, in terms of adverse events, which are toxicities of the treatment, there are very typical treatment-related side effects or unrelated, but the hematological ones are mostly related. So patients developing anemia, neutropenia, or thrombocytopenia, with neutropenia, higher grade being the lead cause here. And this is very consistent with other combination induction therapies. So really not uncommon, and especially not particularly exceeding what has been reported with also commonly used induction regimens at the moment. And then key non-hematological toxicity being infection. Again, that being the commonest cause also with other regimens, and no other specific sign really coming up here that wouldn't be typical for the drugs being used. So in essence, we can say that these results are very reassuring and that no new safety concerns have come up by combining more drugs here than what has been previously tested in other trials. We looked at it the other way around as well. We allowed in the trial to reduce doses of treatments in order for the patients to have a better tolerability of the drugs in the long term. Because we wanted to particularly allow patients to stay on the combination of these multiple drugs for as long as possible. We think that's one of the biggest contributing factors to the treatment being effective for high-risk disease, which always looks for a way of evolving very quickly and effectively being quick at developing resistance against therapy. So if we are attacking it from different angles, the remission time is likely to be longer than if we just use a single drug. And we allowed that because we know certain drugs are having over time accumulated toxicity, and the results here reflect that. Bortezomib, which is known to cause neuropathy, has been reduced in the majority of patients. We allowed to do that early so that higher grade toxicities could be prevented. And I think results reflect that most patients and doctors made use of that. But we also see that other drugs, for example, like Daratumumab, seem to be very well tolerated and have very little cumulative toxicity, reflecting that they didn't have to be reduced, dose reduced, even with long-term treatment post-transplant. So I'm just summarizing here a little bit as a summary of so far, and I will come to then further thoughts that probably will stimulate hopefully a bit of the discussion later on. There's a very clear benefit that we have seen here with this new treatment with Daratumumab CVRD before and after transplant over the comparator data. And that's the first time to our knowledge that this has been shown for a high-risk disease group. There have been other trials. To be fair, there have been few randomized trials so far where patients have had comparison of a new versus an old regimen, but they have not shown a benefit so far. So this is important. The improvement, I didn't mention it much before, but you could see the curves of the two trial outcomes separated the longer time went on. So we think really that the particular effect is also in the post-transplant treatment. To maintain these responses, we did find that there was a high deliverability of this regimen. This trial was performed, especially the second part after the transplant, fell into the first COVID waves, and we had very few people that were not able to continue treatment, which is, of course, on the one hand side, a perfect demonstration of how capable and great the teams have been that have been treating these patients. But I think it's also a reflection on that the types of treatments that we have been using have been particularly suitable, even in difficult circumstances, to allow for continuation of therapy. Safety data is in keeping what we have shown before, but I think the digital comparative framework is really something that is has shown to be a success in that we could find that the two groups are very comparable really in terms of the baseline characteristics. That of course means that we could probably use this as a tool moving forward, particularly to address the high unmet need groups that are rare, that we otherwise cannot benefit that much with the treatment, with the research that we're doing in clinical trials. So there's a patient-centric framework for future research. And I think just really to make it clear, this was a huge collaborative effort. Matthew Jenner, my co-chief investigator, but of course, Sarah's team here, as already mentioned. There have been huge teams in my laboratory. We had a whole team profiling the genetics and the gene expression on an ongoing basis, which we had to, of course, return results, not 24/7, but effectively in really tight time frames. And we managed to do this, which was important. The MRD tests at HMDS Leeds, University of Birmingham, looking at responses. And we also have an imaging component of the trial, which will be done by Christina Masillo and still lead to further results. Of course, the Myeloma 11 team has been really important in delivering this, as well as the trial management group and the UKMRA support, just to name Guy Pratt and Gordon Cook here. But it would have never been possible to deliver this without, of course, our collaborators, both on the treatment and the diagnostic side, our funders that I mentioned here, of course, the Myeloma UK, the David Forbes Six Foundation. We had some support from the BRC and our side for the diagnostic developments, but most of all are really the patients, families, and carers, and of course the investigators and the research teams at the 39 hospitals that you can see here on the right-hand side. So I think I would like to move on now really to some questions. Hopefully we can pick up them, because I think looking at the results now, again, with 1 or 2 months having passed since we presented them at ASH, one can really ask, what has Optimum informed since then? And I think it is quite a impactful research that we have been able to deliver here. So one of the things that we clearly showed was that molecular profiling was feasible in myeloma, with nearly 90% of patients getting a result that gave a sample for profiling. And that was not ever, to my best knowledge, prospectively really shown in a trial. It contrasts with other diseases where, unfortunately, it is far more challenging. There has been a large lung cancer trial, which has been published 1 or 2 years ago, where unfortunately only about 10% of patients even got a molecular result. Now, there are different reasons for that, but it is just showing how feasible it is to really apply very detailed molecular profiling to patients in the clinic. And these results can be actually returned within a time frame where they matter still for adjusting treatments. So this is making molecular profiling really useful based on this data. And this has informed already in the RADAR trial, which is ongoing at the moment, a national trial for about 1,100 patients. That standard of care genetic profiling will be used on these markers, on the double hit markers at least, unfortunately not the gene expression, but we're working with for the next trial on that as well. And these were informed in part by the Optimum definition of high risk, including also deletion 1p, which has not been a standard of care marker in all laboratories so far. And a result is really a motivation that we need to make access to gene expression profiling possible. We do know through the results that there are about 10% of patients that are under or misdiagnosed without such profiling, and work is ongoing at the moment, but we still need, of course, a lot of support to really get this more widely accessible for patients, which it isn't at the moment. What else has Optimum informed? Well, we have found clearly that the addition of an anti-CD38 antibody, in this case it was Daratumumab, but there are others, for example, Isatuximab to induction treatment, improves outcome for patients with ultra-high risk disease as per the standards here. And the RADAR trial actually took this information on board very early and is undergoing an amendment at the moment to include Isatuximab in induction therapy directly from the diagnosis of high risk disease in the trial arm. We have also found that extended consolidation is clearly highly beneficial for patients with high risk disease. Now, Optimum was designed in 2016, and we had long debates at the time. At the time, it was very heterogeneous still, the treatment standard across the country. Patients received CTD, CVD, so only thalidomide or Velcade-based treatment. And we, at the time, advocated already to have a better combination treatment, at least for induction BTD, but we didn't have any way of recommending a post-transplant consolidation therapy. Since then, Revlimid was approved as a maintenance therapy, and we are now fortunate that since January this year, Dara VTD induction and also 2 cycles of Dara VTD consolidation have been approved. But what we have found is that even longer Dara VRD consolidation is really what the patients with these genetic and gene expression characteristics would benefit from most. So that's where we really need to work together on the implementation of the results. And again, efforts are ongoing together with Myeloma UK and other stakeholders that we can hopefully achieve that. One of the design features of the trial was that we designed the trial already with a view to that, when the results come out, nearly all the drugs in the regimen should be generic. Now, whether that means that it makes it more accessible on the system or not is to be seen, but at least the barriers should be far lower than, let's say, if there was a higher cost barrier with several of the drugs still not being in generics formulation. Now, of course, I think what the trial as such has shown, thanks to Thara's team's work in coming up with this digital comparator design, is the value of public research for patient benefit. I think it's really one of the first trials for high-risk disease, and this is something that speaks to the benefits of how we can design research in the UK and how we can deliver research in the UK. But it also shows the value of public data sets for patient benefits. Now, without having run that MUK9 trial previously in the UK, which was also a public trial, if you want so, we wouldn't have been able to actually compare the data against a high quality data set. And I think it really overall speaks for the value of a clinician-patient alliance, as we have here with the CARP model and MUK and the trials unit, but also other stakeholders working very closely together to clearly define and address the needs of patients here. And I think this is a restriction that we find often not by bad intention, but industry trials, they have a different focus. They're focusing often on just getting the licensing of a drug achieved, which is, of course, a huge obstacle, and no public body would be able to do that. It takes huge resources, but it means that there are restrictions inherent on the utilization of this data beyond obtaining a license. And also there are challenges in that the prime market of being considered by pharmaceuticals is the US market through FDA, and that makes it difficult then to actually use results in a different way. Sorry, I have to silence my phone here. Now we have actually other data emerging as well. Sorry for being slightly distracted. I think what is interesting is that we do see context emerging as well from other research that was designed elsewhere. And actually these results are actually very reassuring. So in the US, there was a trial called the MASTER trial, which was designed probably at a similar time point as the Optimum MUK9 trial. And it used also a very intensive treatment combination, DARA KRD, but it took a different approach. It didn't really look primarily at genetic risk. It looked more at response-guided treatment. So patients were receiving minimal residual disease bone marrow biopsy investigations. And depending on the result, on the bottom, on the lower part of the diagram, you could see they could enter a treatment-free observation interval. So if they had 2 subsequent MRD measurements that were negative, they could stop therapy and be observed at that point. And of course, this is a highly, highly laudable principle, and we were, all waiting for the results of this. And but what the authors found and what they presented at ASH as well is that patients who do have 2 or more high-risk markers, so the same definition that we used in Optimum, when even if they had achieved a really deep response with MRD negativity, but then stopped treatment, unfortunately, the disease was far more likely to return relatively quickly than for those that had only 1 high-risk abnormality or no high-risk abnormality. They achieved at the same rate, actually, this MRD negative status. So you couldn't predict just on the basis of how well they responded whether the disease was likely to come back. It was only the information on the genetic markers at diagnosis. And as you can see here at 18 months, although the data is not really mature enough here, it looks like as if about half of the patients have unfortunately experienced a relapse or are going to experience a relapse in that group. Now, this was just a post-hoc analysis. So when they included patients in their trial, they didn't inform them about the genetic status that the patient had. And actually, in the way how they presented, how they had designed the trial, they hadn't even taken the genetics into consideration. But I think the data that was emerging from other studies, including the UK studies, informed them to look in hindsight at this split. And I think what is one of the clearest messages of this, really all patients need comprehensive profiling at diagnosis to advise them on whether it is really making sense for them to go into such a response-driven treatment break, because patients here weren't informed about this. They didn't have a choice to know about this. And I think this speaks to the very first principle of that, empowering patients by generating more knowledge about their disease. And I think this is probably the model that emerges here. This is depth of response here, and in a remitting relapsing disease, depth of response and the speed of the regrowth of the disease matter in terms of what the outcome is in relation to MRD. I think this is not something that you normally see in MRD diagrams. Normally the MRD diagrams suggest the lower the response is, the longer the long-term outcome is. Now, within each group that is correct, but let's assume you have 3 tumors of different risk groups. They reach the same depth of response, but then have different regrowth characteristics. They will have very different outcomes. And actually, we can probably manipulate that. And this is actually, we have demonstrated this now with MUK9. If we keep the patient on intensive ongoing therapy, we can alter the course of that regrowth of the disease, but it needs knowing about this status already in anticipation to react early. Another trial result that was also coming out earlier last year was an interim analysis of the German UKMRA-MUK-Concept trial, actually very similar in terms of really focusing on high-risk disease, but probably a group less stringently defined, so not having as many parameters as the UK trial, and also not focusing on those with the most complicated disease in terms of 2 or more high-risk markers, but patients also received such a 4-drug intensive combination with the CD38 antibody isatuximab. It included also patients that were not fit for intensive treatment, although the results reflect mostly those with intensive treatment, and this included in some even a tandem transplant here. And this is the result of that interim analysis, just the very first 50 patients, which shows that progression-free survival, if you remember the curve that I showed you earlier, that's actually really quite similar to what we have seen in Optimum with a 12-month progression-free survival, slightly nominally below what we have seen with 79% at 24 months, 75.5%. Keeping in mind, this is a more intensive regimen in terms of infusions. Both isatuximab, which is still IV, as well as carfilzomib, require the patient to come up to hospital at least every other week, and with the tandem transplant also requiring 2 longer hospital inpatient stays. But still reassuring in terms of the treatment principle as such, in that prolonged treatment intensity also post-transplant really contributes to improved outcomes in this group. And with that, yes, I often finish my talks with this because I think it just shows how so many pieces of the puzzle are needed to be put together for us to understand what are the elements of treatment that we need to take on board to really target the disease at its driving forces. And I think with Optimum, we have been very fortunate that we have been able, thanks to really sticking our heads together and being very collaborative on a multidisciplinary front to making progress for our patients with high-risk disease. So I'm very happy to speak about these results and take questions. I'll stop sharing my slides at this point.
Thank you, Martin. That was fantastic. Really great to see. Well, obviously great to see the results of Optimum, but also really nice to see them in the context of other trials as well that have been developed and also presented at ASH recently. So if people want to ask questions, please do raise your hand. I think it's the reactions function in Zoom, or type your questions in chat. Just while we're waiting for people to think about questions, I think for me what was really great to see and great to do with MUK9 is the ability of, I think as you mentioned, the collaborative multidisciplinary nature of the study, where it's teams of clinicians, methodologists, scientists, patients working together to develop something, but also the ability that we have in myeloma research in the UK to share the data between the trials. This study wouldn't have been possible in the way that it was delivered without myeloma 11 and without myeloma 9 to help us inform that. And it really highlights, I think, that unique capability of trials that we develop in the UK to really put us at the forefront of research in this setting, particularly when we're dealing with really rare populations. And one of the things I wanted to ask you, it obviously demonstrated a really clear progression-free survival benefit, but you did highlight that it's much more intense treatment. What do you think the implications might be on quality of life for patients?
Yes, it's certainly going to be a very individual result, but I think it has to take into account that in patients that would not receive this intensive therapy, very likely they would have experienced an earlier relapse with a type of treatment that will yet cause different types of potentially cumulative toxicities and impacts on need, for example, to come to a hospital. I think such an intensive regimen inherently always means a certain side effect profile. I think it's very few patients that will have no side effect profile, but one has to keep in mind if with a subsequent treatment, there is probably the side effect profile of subsequent therapy as well as the potential effect that the relapse as such, of course, has on quality of life. And we do know that the disease is unfortunately causing a lot of, ranging from pain to organ dysfunction if it relapses. So I think it's likely that, at least in our assumption, that the quality of life of maintaining a remission, even if there are certain side effects from that, might be beneficial. We will not be able to in detail look into that in the comparison between the two arms, but we have collected very detailed quality of life data from the patients treated in Optimum. So we'll certainly look into that and get an answer at least within that treatment group.
Yeah. Okay, thank you. So we've got a couple of questions in the chat. The first one, what does cyclophosphamide add to the induction regime? So it just says MA, so I don't know if the person who put that in wants to add anything in particular.
Yeah, whilst we may be waiting for that, I mean, it's a difficult question. This has been investigated in other settings. I don't think that it is necessarily so easy for us to tease it out of this here. We do know, of course, that both of the comparator arm regimens, both CRD and KCRD in the myeloma 11 trial, also contain cyclophosphamide. So in some sense, we felt this is actually the fairer comparison if patients also receive cyclophosphamide, and there was a hypothetical consideration as well that there might be types of high-risk disease, and we have seen this actually being confirmed both in our trial as well as in the master trial in the subgroup analysis that remain quite chemosensitive to classical chemotherapy agents. So for example, the melphalan element in our trial has led to an uplift of responses, MRD negative responses, by about 20%, and a very similar rate was seen in the master trial as well. Now, whether that is the same effect that a lower dose cyclophosphamide has as well will be difficult for us to prove, but at least it's a very encouraging thing that maybe these cyclophosphamide has had a similar additive positive synergistic effect to the induction regimen.
Thanks, Martin. Another question that we've got in from Dr. Jim Cavett. So did the plasma cell leukemia patients have different outcomes to other ultra high risk patients?
So this is something that it's probably a little bit too early to make a definitive statement on at the moment. We did have, as you might have seen, spent only relatively little time on that, but in the flow diagram, we did have some plasma cell leukemia patients that unfortunately turned out to very early on already develop resistant disease. These were overall only 9 patients that received treatment with plasma cell leukemia, which is the expected frequency. It shows how difficult it would be to run a trial specifically for plasma cell leukemia alone. I think we made it very relatively easier by combining them with others to actually really address the population as as it emerged throughout the recruitment of the trial. It is still encouraging that the majority of those still had a ongoing remission up until consolidation 1. Whether there's a tiny difference, I think we will need to wait longer, but there's certainly seem to be some plasma cell leukemia patients that still have an ongoing remission, even with the trial being in follow-up now for 2 years longer.
Thank you, Martin. I can't see any more questions in the chat, and nor can I see any hands up. So if I'm missing them, please people do just shout out. But I would just really like to say again, thank you, Martin, for a really great presentation. It's clear that there's lots more to come from MUK9. This is our Optimum. This is the start, really. Lots more analyses to come, and I've made a few notes of things that we can be adding to our analysis plan in the future for us to look at. So it's been really great to see that all together, and we hope to hear some more results in the not too distant future for this. So thank you everybody for joining. The presentation has been recorded, and we will share the link via the YouTube channel. Just before everyone leaves, we do have another webinar scheduled for April. So we're using these webinars at the moment to showcase the results of our trials portfolio within CARP. So in April, Dr. Holger Orner will be presenting the results of the MUK9 trial to us, which is a recently analysed trial that was developed earlier in the Myeloma UK portfolio. So thank you again, everyone, for joining us, and thank you, Martin. Great to hear from you, and we look forward to seeing you all again in the not too distant future. Thanks very much, everyone. Bye.
Thank you. Bye.